see that. You can see that? Yep.
Yes, we can.
Okay, great. Awesome. All right, so thanks so much, and thanks for inviting me. It's great to be here. And so today I'm going to be talking about long-term research and how it has and continues to help us understand turtle ecology, as well as associated and sometimes unexplained behaviors. And so I will be monitoring the Q&A. So if you do have questions throughout the presentation, I can answer them while I'm presenting, and that'll keep the topic, the themes together. And so just a little bit of background. So my work takes place at the Algonquin Wildlife Research Station, which is a field station situated along the Highway 60 corridor in Algonquin Provincial Park. The station was founded in 1944 with the purpose of being an outdoor lab for ecological research. It was first run by the Ontario Ministry of Natural Resources, which was formerly known as the Ontario Department of Lands and Forests. But in the early 1980s, due to some budget cuts, operations of the station were assumed by numerous Ontario universities, such as University of Guelph, U of T, Laurentian University. And since 2010, the station has been considered an independent, not-for-profit organization. And so in the past, the station has hosted research on animals ranging from moose, Algonquin wolves, beavers, but it's now home to quite a few long-term studies, which has hosted numerous researchers like myself. And so the picture on the right here just shows the station cookhouse, which is essentially the station hub where all the researchers go to unwind after a long day in the field. Among the long-term projects is the Bat Lake inventory of spotted salamanders. It first started in 1992 and has been monitoring amphibians that breed within Bat Lake for approximately 30 years. It's one of the youngest long-term studies at the station, and they monitor amphibians that breed within the lake, such as yellow spotted salamanders, blue spotted salamanders, wood frogs, bullfrogs, green frogs, and sometimes tree frogs. Then we have the Small Mammal Project, which is the longest running project at the station and has been running since 1952 for approximately 70 years. And they monitor deer mice, jumping mice, redback voles, as well as red squirrels and chipmunks, and sometimes flying squirrels. And then there's the Canada Jay Project, which has been running for 56 years since 1966. They spend their field season tracking Canada jays or gray jays across the park. And finally, we have the Turtle Life History Project, which has been running since 1972 for approximately 50 years. And so today I'm going to be talking about the Turtle Project, which includes an overview of a typical field season, an overview of turtle ecology and what makes this project unique or important, as well as how the long-term project has and is currently helping us further our understanding of turtle behavior. So Ontario is home to 8 species of native turtles. We have the spotted turtle, the painted turtle, Blanding's turtle, eastern musk turtle, eastern spiny softshell, snapping turtle, wood turtle, and northern map turtle, all of which are classified as at risk in Ontario. Out of the 8 species, 4 of those species are found in Algonquin and are part of the long-term study, including the snapping turtle, painted turtle, Blanding's turtle, and wood turtle. And so today I'm going to be mostly talking about painted turtles and snapping turtles because they're the main focus of the project and are most abundant in the populations that are consistently surveyed. But there have been studies in the past that have focused on Blanding's turtles and wood turtle populations. And so the Turtle Project first started in 1972 by Dr. Ron Brooks from the University of Guelph. At the time, only 20 snapping turtles were tagged. And when I mean tagged, I mean that the turtles were given a unique alphanumerical code and a little metal tag attached to the back of their shell. There wasn't really a goal at the time other than wanting to see if individuals returned year after year. By 1976, 80 females had been tagged, and it was decided that this would become a long-term study of reproductive output and movement. In 1981, it was realized that these turtles were quite long-lived and that this allowed a quite reliable way to measure life history traits and environmental variables in a long-lived species, which complements the long-term study of short-lived small mammals in a comparatively undisturbed and intact aquatic and terrestrial ecosystem. And so in the late 1970s and into the 1980s, painted turtles, Blanding's turtles, and wood turtles were incorporated into the study and monitored year after year after that. And so 50 years later, this project's still running, and it's being co-run by Dr. Jackie Litzkis at Laurentian University and Dr. Niall Rawlinson, my supervisor at the University of Toronto. Many of the turtles that were captured as adults in the early years of the study are still around today and reproducing every year. The study has tagged approximately 1,430 adult painted turtles with 13,323 captures and hundreds of recaptures each year. The study has also tagged approximately 769 adult snapping turtles with 4,085 captures and close to 100 recaptures each year. Our field season starts in May, just as the turtles are emerging from their overwintering spot. Every day we'll go out in the field site, catch turtles from either canoes or from canoes using either dip nets or our hands. These turtles will then be brought back to the station for a yearly checkup. And in other words, they get measured and weighed, and they also get ID'd. Each turtle has a unique ID, which is identified by notches that are on the periphery of their shell, and sometimes a little metal tag attached to the back of the shell. We then paint their ID on their shell and release them back into the water and wait for nesting season. Now in June, nesting season starts. Our turtle team typically splits off into two groups. One group heads off to Wolf Howl Pond to monitor the painted turtle nesting, which is just around Mizzy Lake Trail if you've been to Algonquin, and the other group stays around the research station and monitors parts of the highway for nesting snappers just around the Lake of Two Rivers area. Once a clutch of eggs is laid, we'll then dig up the nest, measure and weigh the eggs, and either rebury the nest 24 hours later or bring the eggs back to the lab for various projects. With early mornings, late nights, and peak bug season, this time is probably one of the most exhausting periods of our field season, but it's also one of the most rewarding times. There's nothing cooler than seeing your first turtle lay her nest and seeing her eggs drop into the cavity one by one. And so after nesting season, we may do some trapping in nearby lakes in order to capture the adults that we may have missed during May and June. We don't always trap in July, and this varies year to year. For example, this year we didn't trap in July, but one of the grad students came back this August to catch males and get a more thorough sample of the males in the population. And then finally, in late August and September, our turtles start to hatch. Depending on the year and the specific student project, we may monitor turtle eggs in the lab, which is shown in the left photo with all the hatched painted turtles, or in the field, such as the Lake Saskatchewan Dam, which is just on research station property. And so all turtles are ectotherms or cold-blooded, meaning they can't generate their own body heat and need external heat for all physiological functions, such as moving, eating, and mating. Ectotherms can't survive in very cold climates, and so they're not found very far north. And so one thing that's particularly unique is, and what makes the Turtle Project special in comparison to long-term studies that may be taking place more south or in other parts of the USA, turtles in Ontario are at their northern range limit and are thermally constrained by these cold temperatures and short growing season. The Algonquin study in particular is situated at one of the most northern points of their range. Some turtles do live at higher latitudes than Algonquin, but Algonquin has a high elevation, making the climate similar to climates at more northern latitudes. This creates the perfect study systems to study their behavior, physiological processes, especially when comparing them to southerly neighbors. And so many turtles exhibit temperature-dependent sex determination, meaning that the temperature that the clutch or their eggs are incubated at determines the ratio of females to males produced. So in painted turtles, cooler temperatures will produce more males, while warmer temperatures will produce more females. And then in snapping turtles, it's a little bit different, where intermediate temperatures will produce more males, while extreme cool and warm temperatures will produce more females. For example, in 1983 and 1984, naturally incubated painted turtle nests were mostly female biased, which is shown in blue, where survival was relatively high and the proportion of males to females is quite low. This was in the 1980s, and so you can imagine that the impacts of climate change, warming temperatures, and greater temperature fluctuations could potentially have grave effects on turtle populations. In fact, when you compare female producing nest temperature profiles in Algonquin, which is just shown in green here, to female producing temperatures in Iowa, which is shown in red, and Nebraska, shown in black, Algonquin nest temperatures are characterized by a wider fluctuation of temperatures than both Iowa and Nebraska. And so this study in particular used Ontario nest thermal profiles to test how painted turtle eggs from Iowa would behave under future climate change conditions. And so under these future conditions, which is expected to have wider temperature fluctuations, we may expect there to be fewer male producing temperatures and mostly feminizing temperatures. So essentially meaning that clutches would be 100% females. So these, in addition to that, these high temperatures could potentially increase mortality due to being too hot and possibly accelerate demographic collapse in turtles with temperature sex determination. Now, what makes the study system even cooler, turtles are really long-lived, so they can most likely live past 100 years. But that also means that they're more likely to outlive their researcher. And so this research allows us to track individuals over time and confirm whether they're still alive, as well as their growth over time. When it comes to trying to age a turtle, it can be quite challenging. Theoretically, you could count the growth lines on the shell and get an age estimate that way, sort of like counting the growth rings on a tree. However, turtles spend most of their life underwater, which means that over time, the growth rings erode and become quite challenging, if not impossible to count. On the left picture, you can see the really nicely defined scutes and the growth rings in the shell, where you could potentially age the turtle quite accurately. And on the right, you have a turtle that's been underwater quite a bit, and the growth rings are almost indistinguishable. Some may think that you could age a turtle by their size, and while a size age estimate may be a little bit more reliable in the early years when growth is really rapid, it simply isn't accurate. So for example, turtles that are 25 centimeters that have a shell length of 25 centimeters may be 20 years old, or they may be 60 years old. It's a pretty wide gap of 40 years. Or a turtle that's 50 years old could be 20 centimeters, or could have a shell that's 20 centimeters in length, or a shell that's 35 centimeters in length. And so by keeping track of these turtles year after year, we're able to learn more about their life history, including aging. And so in addition to living a long life, this life history trait comes with the fact that it takes quite a long time for turtles to mature. A female snapping turtle may take 17 years before she lays her first clutch, where other species in the park, such as black bears, moose, and white-tailed deer, may take 6, 3, or 2 years to produce their first offspring. So to put that in perspective, by the time a turtle produces one clutch of eggs, black bears would have replaced themselves 7 times, moose 303 times, and deer 629 times. This is assuming mortality doesn't occur. When we take mortality of offspring into account, it can take decades of nesting for just one egg to survive and to replace the turtle that laid it. It's been estimated that it would take a snapping turtle around 59 years to replace itself in the population. Turtle eggs and hatchlings are extremely vulnerable, so nests are easily found and destroyed by predators such as foxes, raccoons, ravens, martins, minks. And the hatchlings that do survive incubation are vulnerable to both predators on land and in water. However, if a hatchling survives until maturity, they have a higher chance of survival. So adult turtles are quite hardy and are practically built like miniature tanks. So they have very few natural predators and can survive decades. Of course, they aren't invincible and will sometimes be killed by river otters while they're overwintering at the bottom of lakes or ponds or swamps. And so winter is a time where turtles are most vulnerable, or adult turtles are most vulnerable, because they essentially do not have enough heat or energy to move and must really rely on the spot they chose to bury themselves in. And so in the late 1980s, snapping turtles from the long-term study experienced an acute mass mortality event. So during the winters of 1986 to 1989, approximately 40% of the adult population of snapping turtles were predated by river otters. And so continued population monitoring not only allowed us to follow up almost 25 years later, we saw that even though there was a return to high survivorship and enough time for offspring that were born after the catastrophe to start being recruited, the population has yet to recover from its original abundance. And so while this is a very natural event, it serves as a wonderful case study for the importance of conserving adult populations of turtles. Mass mortality events can have grave repercussions in long-lived species, where turtles are unable to compensate for declines in their abundance. And so when you apply this knowledge to anthropogenic threats, the impacts are massive. In addition to threats like habitat destruction, pollution, and environmental change, threats like hunting, poaching, and road mortality can have long-lasting effects. When you think of Southern Ontario, you can't get anywhere, or within 1 kilometer, you're going to encounter a road. And so roads are everywhere, and turtles eventually will have to cross them. And so it's quite a big threat, and reason why it's important to mitigate these threats. But on the happier side, long-term research at the station has helped us towards protecting turtles and preventing them from further loss. The Turtle Project has helped inform both policy and conservation action. So recently, information from the turtle study helped inform the decision to add painted turtles to the list of at-risk turtle species within Ontario. And some of the people who wrote the report or helped write the report came from the turtle study. And so that made the painted turtle the last native turtle species to be designated as at risk. Similarly, the turtle study massively informed the snapping turtle Kasiwik report. And so this resulted in delisting the snapping turtle as a game reptile and the end of recreational hunting of snapping turtles in Ontario, which could very much help the population from further loss. And so in the past, research on the Turtle Project has also focused on applied conservation. Some research in the past has evaluated the effectiveness of different types of nest cages and turtle hatching success. A lot of concern with nest cages originally was that perhaps nest cages would alter the thermal conditions within the nest. And so this study showed that it doesn't matter what nest cage you use, it prevents predation, but it doesn't alter the thermal properties. And when you look at the predator interactions with these nest cages, we see that predation occurs both at the beginning of incubation when the nests are laid and at the end of incubation when the eggs are close to hatching. And it was previously believed that predation only occurred really in the first few weeks of nesting, when the olfactory cues or the smell of the nest would be really strong, and that you would only need to protect the nest for a few weeks for it to survive predation. But this study really helped us understand that predation occurs throughout the season. Although the reason for the second peak late in the season is unknown, it's better to leave the nest cage on for as long as possible. And to this day, we still use these cages to protect turtle nests. And so to summarize, the turtle study has investigated growth, reproductive output and success, recruitment, genetic variation, movements, diet, impacts of increased mortality, life history changes, and the role and effects of thermal constraints at their climactic range limit. And we now have a unique data set spanning over 50 years and thousands of individuals and their responses to climate and demographic pressures. And so these long-term studies are essential to understand such phenomena, as well as the formulation and testing of ecological theory. The Turtle Project has nearly published 100 scientific papers. And so to talk about all of it would be a huge task. There's so much that has been done. But what's amazing is that it's hosted quite a few grad students, so about 25 grad students, and it's trained and introduced about 100 undergraduates to the field of biology, really educating and helping train the next generation of biologists and field biologists. Despite all we know, there's still a lot to learn. So even though there's been 100 papers that are published, there's still many questions to be asked. And that it relates to turtle reproduction, turtle communication, sociality, nest site selection. But one thing that's great about these long-term research projects is that there's room to conduct short-term studies, and many of which help us understand these lesser-known phenomena and behaviors. And so perhaps some of the more simple behaviors include dispersal and migration. Turtles are quite mobile. Some are more mobile than others. So the blanding turtle may travel up to 6 kilometers to find a nest site and travel 6 kilometers back to overwinter. compared to painted turtles who may travel about 3 kilometers. But generally, turtles move around quite a bit. And so in the spring, turtles will wake up from their overwintering spot and start to forage for food after a long winter nap. And then in the early summer, turtles will start to mate and will eventually migrate to lay their eggs. Then in the fall, turtles will migrate once again to find a spot to overwinter. And of course, this varies between species. Painted turtles will sometimes lay 2 clutches of eggs, so they'll go through 2 mating seasons, and the snapping turtles will only mate once and lay 1 clutch of eggs. Snapping turtles in particular will travel on average 16 kilometers round trip just to migrate to their nest site. And so in this figure here, you can see that each green circle represents a turtle who was found in the early spring or by trapping, all of which nested on the Lake Saskatchewan Dam, which is marked with a red star. It's also shown in the picture on the right. And so why are snapping turtles choosing to travel up to 8 kilometers just to nest on the embankment of Lake Saskatchewan? For a turtle that's only about like a foot long, that's quite a bit of distance to travel. One idea is that these turtles are taking care of their eggs and choosing a site that will give their offspring the best chance of survival. Compared to randomly selected sites, painted turtles will select sites with little canopy cover, little vegetation cover, and a southwestern facing slope. This means that nest sites chosen by turtles are at a location that's optimal for their offspring and will have the highest chance of receiving enough heat to develop over the summer and hatch before winter arrives. Many believe that reptiles like turtles don't care for their young, but this suggests that, at least in part, turtles take care of their young. I will mention that there are some turtle species. Specifically one, a South American river turtle, who actually do exhibit more elaborate parental care, and they take care of their young by waiting for their young in the water body and waiting for the young to emerge from their nest, and then they all migrate together. But this is just a behavior that hasn't really been observed or tested in Canada's freshwater turtles. And so what makes the Lake Saskatchewan Dam so special? Wouldn't there be sandy? You would think that there might be sandy slopes somewhere upstream, a little bit closer to the overwintering site. Better yet, why do you most often see turtles nesting on roadsides if it puts them at risk of being hit by cars? What makes sites like the dam and roadsides so special? Well, they have one thing in common. They were both created by man. In fact, man-made substrates tend to be thermally optimal and preferred nest sites by turtles. Anthropogenic nest sites have less ground level vegetation, greater soil brightness, and are approximately 3 degrees warmer than natural nests. Because of this difference in temperature, it's predicted that eggs in anthropogenic nests will develop nearly twice as much, meaning that their offspring have a higher chance of leaving the nest before winter. And we can see the difference in this figure, where the top row shows the natural nest sites where you have more dark, organic, rich soil. One of the nest sites is in a beaver hut, and so that is quite dark soil. And then the bottom is the man-made sites, which have these pale, sandy soils. Whether it's actually beneficial for turtles to nest on these anthropogenic nest sites is still unknown, but they appear to prefer the thermally optimal temperatures and substrates. And so, as I mentioned earlier, turtles are ectotherms, meaning they don't do well in cold environments. So not only do turtles have to survive cold temperatures, they have to survive at the bottom of lakes that receive very little oxygen circulation in the winter months. Despite this, we still see turtles thriving in the cold and north parts of Ontario. A lot of this has to do with their ability to choose the habitat where they overwinter. After nesting, they'll travel many kilometers to reach their home lake. And turtles like the Blandings turtle and painted turtle will often choose to overwinter in habitats like bogs, fens, swamps, and ponds, largely because these regions are more thermally stable. And so this graph is just an example showing a Blandings turtle's body temperature as they overwinter compared to temperatures at random sites within a marsh environment. And so the turtle body temperature tends to have less variation than the marsh temperature. which is quite variable. And so a lot of research on reptile or turtle behavior often relates to factors like habitat choice, movement, dispersal, or physiological processes. But when it comes to the study of social behavior, which is the study of the interactions between individuals of the same species, reptiles are often neglected, especially in comparison to studying social behavior in mammals and birds. Reptiles aren't traditionally seen as charismatic and are recognized as sort of these cold-blooded or cold-hearted organisms. And so the notion of complex behaviors like parental care and social groups or even mating are less recognized. Because of this, research on reptiles, and in this presentation specifically, turtle social behavior is quite new. Despite previous beliefs, turtle social behavior can be quite complex. So take the painted turtle, for example. Traditionally, everyone sees the painted turtle as this perfect gentleman where males will court females using their long fingernails and engage in a behavior called titillation, essentially tickling the females with their fingers to convince the female to mate with them. Between males and female painted turtles, you can see this sexual dimorphism where females are often larger with these domed shells and short fingernails, and males are often smaller, a little bit more flat with longer fingernails. While this courtship behavior does occur, it's not the end of the story. Females may choose to mate with these young males, but as the males grow and become less appealing to females, the males switch from being these nice gentlemen to having more aggressive advances on females. In fact, male turtles have these enlarged tooth-like cusps at the front of their beak, as well as these sharp projections from their shell. And so they essentially use these weapons to mate with females. You can often see that females have a disproportionate amount of injuries from the aggressive advances. These injuries are usually found on the top of the head and neck, which is consistent with the regions that males would bite. And so males will often match their strategy to their morphology, where large males will show aggressive mating behaviors and will use their sharp projections from the shell and beak, where small males will use their long fingernails and be a little bit nicer and court the females. And I'm just going to answer a question. So Sean mentioned, couldn't the choosing, so this relates to nest sites. So couldn't the choosing of the anthropogenic nest sites versus natural sites change the male to female ratio as per your comment about temperature changing the sex of hatchling? And could they be doing this on purpose to guarantee the number of each sex produced as necessary for ongoing survival? Yeah, so a lot on, so this is a question, so yeah, sex ratios is a big topic within the lab group. I don't specifically study temperature sex determination, but yeah, by nesting in anthropogenic nest sites, it essentially could skew the female to male ratio. Whether they're doing it on purpose and it's a choice that they're making, it's possible. Most often, I would say that the turtles are choosing it based on thermal properties and hatching success, and perhaps less on the sex of each turtle, the sex ratio of the clutch. Yeah, and so in terms of, and so could they be doing this on purpose to guarantee the number of each sex produced as necessary for ongoing survival? So to answer your second question, we don't actually know whether anthropogenic nest substrates are more beneficial for nest sites. And so we aren't sure whether it is an ecological trap or if it's a, if it's an ecological trap or if it is actually better for the species. And this is a topic that is of interest. And I know one student may be doing a small research project on that. But yeah, we don't know. Might the painted numbers on turtles perhaps cause turtles to be hunted more successfully by humans? Yeah, that's a great question. That's a question that we get a lot. So at Wolf Howl Pond, which is where our painted turtles are at, we paint the backs of the shells with red or white paint. So red means it's a male, white is for female. And we do this so that we can easily identify the painted turtles when they come up to nest. And so in terms of being hunted by humans, probably less so. So we are all in the protected area of Algonquin Park, and so there isn't any human hunting. But I have gotten comments before of whether this impacts predation and whether there's more predators. And so in the summer months, adults don't get predated, and the paint ends up washing off every year because they shed their scutes every year. In terms of younger turtles, so hatchlings, we don't paint them. So we only put a really small dot so that we know that we caught the individual, but we don't put a huge letter and number combination. And that's so that predators do not identify them more easily. But that's also, we don't actually know whether that, we don't actually know whether that increases predation. But yeah, no, great questions. So I will move on. And so one of the topics that I find really interesting, and the topic, so the topic that's central really to my master's and that I'm working on is nest sociality. All turtle eggs are laid in groups. However, these eggs most often don't develop at the same pace. The eggs at the top of the nest experience warmer temperatures with high fluctuations in temperature, where eggs at the bottom exhibit these cool temperatures with low fluctuating temperatures. And so you may expect these turtles to emerge asynchronously, where the ones at the top leave the nest first because they have a faster development than the ones at the bottom. However, turtles often hatch at the same time as their siblings. And sea turtles, especially, you often see these large mass emergences on beaches. Perhaps freshwater turtles don't really come out of their nest in such large groups like sea turtles, especially when you compare a sea turtle nest, which has 200 eggs. If you compare that to a painted turtle nest with about 8 eggs, or a snapping turtle nest, which has about 20 to 60 eggs. But the truth is that very few people have really studied freshwater turtle emergence. There's only been a few species that have been studied. And so compared to other reptiles, we know a lot about turtle nests. Turtle nests are one of the easiest reptile nests to find and study. However, no one really knows what cues emergence, and not many people have quantified whether freshwater turtles synchronize nest emergence or come out of the nest as in sync as you see sea turtles coming out of the nest. And so painted turtles specifically, painted turtles are quite interesting because they have 2 different types of nest emergence behaviors. Some nests will emerge in the autumn, while others may choose to emerge from their nest in the springtime. Painteds are particularly good at withstanding freezing temperatures and can survive nest temperatures during the winter, in comparison to snapping turtles that can't survive freezing temperatures and will mostly emerge in the autumn to overwinter elsewhere. No one's really sure what cues autumn versus spring emergence, but some previous work at the station suggests that it has to do with environmental factors, where smaller turtles will emerge in the fall, possibly to increase the amount of energy reserves needed to overwinter. So they're much smaller, but the idea is that they're foraging before they go to overwinter to sort of bulk up and get more energy. But another possibility is that these turtles are emerging early in the autumn to avoid predation from flesh fly larvae. But again, this is more observational and there hasn't been specific tests. And so in snapping turtles and other Ontario turtles, we know even less about their emergence. We know that hatchlings emerge in the fall, but not much research has looked into what cues emergence or hatching. And so my research is focused on understanding why turtles leave their nest together and whether this has to do with communication among the eggs. Specifically, I'm interested in understanding whether acoustic communication or vocalizations, the sounds that turtles make, are involved in hatching and or nest emergence. Now, when you think of vocalizations, you think of bird songs, a dog barking, maybe a wolf or coyote howl. And so studying reptile sounds is quite unusual. Reptiles aren't really known to make sounds or vocalize. And so a lot of what we know about reptile vocalization stems from research on alligators and crocodiles. This group was first suspected to vocalize in about 1915. It wasn't until the late 90s and early 2000s that we understood that these vocalizations function in social behaviors like hatching, parental care, and even courtship. It took quite a bit of time before we were able to figure out why these crocodilians were producing sounds. Now, turtles are similar to crocodiles in the sense that they also vocalize, which I alluded to previously. We now know that there are more than 50 species that vocalize. However, we still don't know much about why they vocalize. Like crocodiles, some research suggests that these vocalizations are especially relevant for hatchlings. And so my interest in this topic really started with an undergrad thesis. I was interested in understanding if snapping turtle hatchlings vocalize. It was a pretty simple project involving a simulated turtle nest where we buried the eggs in an aquarium in sand, as well as we buried it with a microphone and a camera to indicate when the turtles were hatching. Little did we know, snapping turtle hatchlings produce up to 5 different types of vocalizations. And so we have the type 1 vocalizations, which are very simple and they're very hard to hear. We have type 2 vocalizations, which are a little bit longer, but again, still quite simple. Then we have type 3 vocalizations, which have about quite a bit of pulses, and it's a much longer vocalization compared to types 1 and 2. Then we have type 4 vocalizations, which are more like a squeak, and they have, what's really interesting about these vocalizations is that they have these clear harmonic bands in these spectrograms. So in the little blue box, you can see these nice red and almost teal-like bands, and those are called harmonics, and it's what you often see in bird songs. And then we have type 5 vocalizations, which again have these really nice harmonic bands, but this time it's at a lower frequency and it's a longer duration. And so by comparing vocalization count over each behavioral period, we observed that vocalizations were more frequent and more complex after hatching from the egg. So before pipping, we only see type 1 vocalizations being produced. And in the latter half, we see that the more complex types 2 to 5 are produced. And so this tells us that it's possible that more complex information may be exchanged among siblings during pipping and emergence. And so why are snapping turtles vocalizing in the first place? Well, it's very likely that the vocalizations function in synchronized emergence or hatching. If this is true, then we may expect there to be an overall energetic or some sort of evolutionary benefit to nest sociality, which would provide more energy to either possibly escape predation or find a suitable overwintering site to forage and find a suitable overwintering site. And so I set up a complementary experiment and tested whether there's a benefit to hatching in these groups. I used a similar setup as the aquarium, except this time I buried eggs in jars of sand. I buried them in either a group of 10 eggs or a single egg. I found that the group of 10 eggs were often the first to hatch with respect to each clutch, and the hatchlings that hatched alone hatched significantly later. This is especially cool because it indicates that siblings have some sort of cue that modifies hatching behaviors. So in every clutch, we saw that the group of 10 eggs were quite low, or they were significantly lower, and they hatched quite a few, or about I believe it was 1.5 days earlier than the clutches that were, or the eggs that were alone without their siblings. I also found that the combined digging effort from the group of 10 hatchlings resulted in less weight being lost as they climbed out of the nest. So which overall means that there's more energy available for other activities like avoiding predators or finding a suitable overwintering site. In the graph here, you can see the dark green. Bars represent the clutch with the 10 eggs. We see that they are both, doesn't matter what nest depth they were buried at, they were both lower than the eggs that were hatching alone. And so these two experiments brought up more questions. Couldn't the turtles hear the noises? Oh, so we have a question from Amelia. So couldn't the turtles hear the noises from the jars next to their jar in your study? Yeah, so that's a great question. So the jars were quite close together, but the plastic's pretty thick. And the other thing about hatchling vocalizations is that it's quite a low level. So it's a very quiet, noise. And so when you add background noise from inside the nest, so the sand makes quite a bit of noise, and you add even background noise if you add background noise in the lab or just white noise, it drowns out the sound. So you wouldn't be able to hear it with the human ear. And even, let's say they did hear it, we still saw a pretty significant effect between the two treatments. But yeah, no, that's a great question. And so yeah, these two experiments brought up more questions. What cues hatching? Why are hatchlings vocalizing? Clearly, there's cooperative behavior going on, but I couldn't help but wonder whether vocalizations played a role in it. So even though I looked at vocalizations and nest sociality, I didn't compare the two. They weren't combined. So another question from Rosemary. Have you used an external device that can monitor these egg sounds in nests in the ground or in an outdoor setting? That's a great question. I will get to that. I will get to that. Yeah, so this brought up more questions, and so I decided to run a third experiment. I took the experiment one step further and tested whether vocalizations cue hatching. I placed earphones next to eggs and played a clip of vocalizations, white noise, or no noise to see if eggs that receive a vocal cue hatched earlier than the eggs that receive no cue. So do vocalizations cue hatching? My experiments suggest that they don't. We saw no difference in hatch date in either of the treatments. This is shocking because many researchers thought that there would be an effect. And so perhaps vocalizations are used in other behaviors such as coordinated nest emergence or post-nest emergence behaviors. And that's why for my master's, I'm going to be recording vocalizations in a wider array of species across Ontario and further investigating these vocalizations and associated behaviors. And so one project that I'm currently working on is characterizing the vocal repertoire of all of Ontario's turtle species and some additional exotic species. Currently, I'm collaborating with organizations across Ontario to analyze blandings turtles, wood turtles, soft-shell turtles, musk turtles, map turtles, and painted turtle vocalizations. I then plan to compare the vocalizations to see if there's a correlated evolution with life history traits such as body size and the average clutch size. So for example, if hatchlings vocalize to synchronize nest emergence, then potentially turtles with large clutches may be more likely to vocalize. This is because there might be a greater need to communicate and coordinate emergence. In addition to that, we may also expect the complexity of vocalizations to increase with clutch size. So we may expect more harmonics, we may expect these more elaborate vocalizations. And this is with the idea that perhaps more complex vocalizations means that there's more information being transmitted. Now, of course, that's a more theoretic approach. And so this past summer, I set up microphones in wild snapping turtle nests in order to test whether the amount of vocalizations relates to synchronous nest emergence. And so to answer Rosemary's question, which is, have you ever used any external device that can monitor these egg sounds in nests in the ground or in an outdoor setting? Yeah, that's essentially what I'm doing now. So I have set up, we have about 17 or 19 nests out on the dam, Lake Saskatchewan Dam. And I dug down to the bottom of these nests and I put in microphones in each nest. And so right now on the left, you can see the nest cages. which have snapper nests all underneath. And then on top, I have a camera trap and a microphone set up. The microphones are just in the little white box on the dam. And then the picture on the right is one of the nests when I was digging down to add a microphone. And this is with the idea that perhaps nests that emerge more synchronously, or that take less time to fully emerge from the nest, have a higher rate of vocalizations. And so right now I'm in the stage of data collection. And so these recorders are doing a lot of heavy lifting, recording a lot of hours of hatching eggs. And then the cameras are doing the work on the top, monitoring when these turtles are coming out of their nest. And then later on, I'm going to be comparing those results. Yeah, so I don't have any results for now, but yeah, it's a pretty cool project. And so, yeah, thanks everyone for listening. And I'd just like to acknowledge that this work wouldn't be possible without the ongoing supervision and collaboration with Christina Davey and Niall Rawlinson, and the support from the Rawlinson Lab. And thanks so much. So happy to answer any other questions.
Okay, I'll just wait for anybody who wants to ask a question. That was fabulous. Claudia, thanks so much. Very interesting research. I haven't heard a lot of stuff. I'm sure some people learned a bunch of things from there. There's a question for you.
All right, so there's an anonymous attendee. Are you looking for more students? Yeah, so I want to say it's not really my job. So Niall, who's my supervisor, he is the one who will hire grad students. And so for undergrads, he takes on thesis students from U of T, but he'll also sometimes take on volunteers. It's more about reaching out to either Niall or yeah, it depends on what you're interested in. And there's U of T students probably have more course-based opportunities, but then you never know what works might be available. Yeah. And so Rosemary asked, where will you publish your results? Yeah, so the results that I just showed, we just submitted to Animal Behavior. And so it's just, right now it's under review. So we don't know if it will be published there, and we're just waiting for the decision and results of that.
Okay. That could be it. There may be a couple more in there, but we'll probably call it a night. One thing I was interested, didn't know about the wood turtles hibernate in water. I didn't know that one.
Yeah.
Kind of interesting seeing that image of them there underwater.
Yeah.
That was interesting. Oh, there you go. There's a couple more.
Okay. Just a comment. You were very thorough in your discussion. I learned a lot. Very interesting. Thank you very much. Oh, thanks, Lynn. And Amelia also said thank you. That was very interesting. Okay. Catherine asks, when you have eggs in bottles, how do they get oxygen? Yes. So I actually drilled holes into the back of those bottles to increase airflow. But another thing to consider is it might have not been necessary because the jars aren't airtight. And one factor that is probably more important when it comes to egg incubation, and one thing that we realized in the lab, is that when you add holes, you lose moisture. And so even though I added those holes, I think keeping moisture was probably more important, but they still hatched, and it was a pretty high success rate in hatching. Marika asks, if someone has a degree in the physical sciences, might we be able to volunteer at your center? Yeah, so there's, at the Algonquin station, sometimes there's volunteers. I want to say there's not too many volunteers, and it depends year from year. Sometimes there's postings for, like, let's say the Canada Summer Jobs Program, and it depends on the project. I would suggest following the Algonquin Wildlife Research Station. They put up a lot of opportunities at the station on that website. And so on their Facebook page, as well as like mostly their Facebook page. So that would probably be the best place to look. And physical sciences, it doesn't usually matter, as having a background in science is good, but even if you don't have a background in science, it's really about being interested. Okay, so another question. My question would be, if you know that there are eggs and they do not hatch by late September, would it be a good idea to just take them out and put them in an incubator? As you mentioned, the chance of survival is low during winter. Yeah, a lot of nests don't survive through the winter, and a lot of nests fail to hatch. And by, I guess, the time that it's a little too late to hatch, it's possible that the temperatures would have killed the eggs, or the eggs wouldn't have been viable. So technically you could put them in an incubator, but the chances of them surviving after incubating for so long, probably low. It's better to just let them be in their nest. These turtles have survived for like decades, if not centuries, and it's okay to leave the nest in. Some programs do head starting though, but those are more under permits, and there's a lot of permits and animal care considerations that go into that.
Okay, I think that's, oh, okay, another, thank you, that's great. Yeah, that was just fabulous, just great. And I'm sure a lot of people got a lot out of it. So thanks very much for taking the time to present to us. And we'll be looking for interest for more details on your work and where it is published and see what's happening. So just see how the turtles are doing in the long run. Thanks,
Awesome.
Claudia. Thanks everybody for coming, and we'll see you if you're going to be at the bird study meeting next week. If not, we'll see you at the AGM on the 18th. Take care and have a great month. Bye all.